Higher Level Biology for the IB Diploma Programme, Pearson book, 3rd edition. Theme D, Continuity and Change, D1.1 DNA Replication. Guiding questions: how is new DNA produced? How has knowledge of DNA replication enabled applications in biotechnology? When the molecular structure of DNA was first presented by Francis Crick and James Watson in 1953 in the scientific journal Nature, it immediately stimulated research into the process of DNA replication. The production of DNA has to be accurate so that new cells or organisms could continue the functions of life. Just as important, this production of genetic material has to allow change, mistakes, if you prefer, to account for the variation obvious in organisms over time. Knowledge of this replication process has resulted in tremendous advances in biotechnology. We are seeing applications of DNA replication in many areas of medical science, as well as in police investigations involving forensics. DNA analysis has led to revelations concerning family ancestry, as well as predictions concerning the inheritance of medical conditions. By understanding the DNA production process, scientists have been successful in replicating extremely small amounts, and in some cases very old samples of recovered DNA. We have now sequenced the DNA of a 1,000,000-year-old woolly mammoth and extracted cartilage from a 125-million-year-old Caudipteryx dinosaur. If DNA can be recovered from this cartilage, it might actually be possible to sequence a dinosaur's DNA. D1.1.1, the role of DNA replication. DNA replication as production of exact copies of DNA and identical base sequences. Students should appreciate that DNA replication is required for reproduction and for growth and tissue replacement in multicellular organisms. DNA structure is discussed in Chapter A1.2. It is a polymer composed of monomers called nucleotides, bonded together. Each nucleotide includes a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. There are 4 nitrogenous bases in DNA: adenine, thymine, cytosine, and guanine. The basic structure of DNA is shown in Figure 1. Note that the bonding of adenine and thymine involves 2 hydrogen bonds, while the bonding between cytosine and guanine involves 3 hydrogen bonds. The outside strands are of alternating phosphate groups, shown as circles, and deoxyribose molecules represented by shaded pentagons. DNA provides the code that creates the uniqueness of all forms of life on our planet. This code is carried in the sequence of the 4 possible nitrogenous bases. Because DNA is the code of life, it must be duplicated so that the reproduction of organisms is possible. DNA duplication is also essential for growth and tissue replacement in multicellular organisms. This duplication is known as DNA replication. Replication doubles the quantity of DNA and ensures that exact copies of each DNA molecule are made. In 2014, a group of Scripps research scientists, so Scripps Institute is a research institute, introduced 2 new unnatural nitrogenous bases into the genetic code of living bacteria. These new bases were incorporated into the DNA code for the bacteria. The research is ongoing. Many feel that this approach could provide a means by which new medicines and vaccines can be developed. With 6 bases instead of 4, many more products could be produced. D1.1.2 and D1.1.3, semi-conservative replication and complementary base pairing. D1.1.2, semi-conservative nature of DNA replication and role of complementary base pairing. Students should understand how these processes allow a high degree of accuracy in copying base sequences. D1.1.3, role of helicase and DNA polymerase in DNA replication. Limit to the role of helicase in unwinding and breaking hydrogen bonds between DNA strands and the general role of DNA polymerase. The replication of DNA is said to be semi-conservative. Each strand in the DNA double helix acts as a template for the synthesis of a new complementary strand. Evidence for a semi-conservative replication process was provided by research using bacteria. See Figure 2. A diagram demonstrating the general process of semi-conservative replication of DNA. You can see the parent DNA, the products of first replication, and products of second replication. In the 1950s, Matthew Meselson and Frank Stahl carried out research using radioactive nitrogen that provided evidence for the semi-conservative replication of DNA. See the ebook for an activity. Figure 2 shows how each daughter DNA double helix contains an old strand from the parental DNA double helix and a new strand, thus the name semi-conservative replication. In the nucleus of cells are two types of molecules that are particularly important for the process of DNA replication. They are enzymes needed for replication, which include helicase and a group of enzymes collectively called DNA polymerase. Free nucleotides, which are nucleotides that are not yet bonded and are found floating freely in the nucleoplasm. Some contain adenine, some thymine, some cytosine, and some guanine. One of the first stages in DNA replication is the separation of the double helix into two single strands. You should remember that the double helix is held together by the hydrogen bonds between the complementary base pairs, adenine and thymine, and cytosine and guanine. The enzyme that initiates this separation into two single strands is called helicase. See Figure 3. The first step of DNA replication is helicase, unzipping the double-stranded DNA molecule, forming a section with two single strands. The unpaired nucleotides on each of these single strands can now be used as a template to create two double-stranded DNA molecules identical to the original. Some people use the analogy of a zipper for this process. When you pull on a zipper, the slide mechanism is like helicase. The separation of the two sides of the DNA molecules is like the two opened sides of a zipper. The process of semi-conservative replication, as described, allows a high degree of accuracy in copying base sequences and provides stability when passing on the genetic code. Formation of two complementary strands. Once DNA has become unzipped, the nitrogenous bases on each of the single strands are unpaired. In the environment of the nucleoplasm, there are free-floating nucleotides available to form complementary pairs with the single-stranded nucleotides of the unzipped molecules. This does not happen in a random fashion. A free nucleotide locates one end of an open strand, and then a second nucleotide can arrive to join the first. To join, these first two nucleotides must become covalently bonded together, creating the beginning of a new strand. The formation of a covalent bond between two adjoining nucleotides is catalyzed by one of the DNA polymerase enzymes, which is an important part of the process. A third nucleotide then joins the first two, and the process continues in a repetitive way for many nucleotides. The other unzipped strand also acts as a template for the formation of a new strand. This strand forms in a similar fashion, but in the opposite direction to the first strand. In figure 4, notice that one strand is replicating in the same direction as helicase is moving, while the other strand is replicating in the opposite direction. Figure 4, DNA replication showing the enzymes helicase and DNA polymerase at work. Helicase is opening the double helix, while DNA polymerase is catalyzing the formation of covalent bonds between adjacent nucleotides as complementary base pairing occurs. The significance of DNA replication is that it ensures two identical copies of DNA are produced from an original strand. Notice that in the area where replication has already taken place, the two strands are identical to each other. This is because the original double-stranded molecule has complementary pairs of nucleotides, and it was complementary nucleotides that used the unzipped single-stranded area as templates. There's a linking question here. How is genetic continuity ensured between generations? You can use different areas of the book, different concepts to think about that question. Helicase has been found to catalyze the unzipping of DNA at a rate measured in hundreds of base pairs per second. That is very fast. The role of helicase in DNA polymerase in DNA replication. The enzyme helicase is essential during DNA replication. It separates the double-stranded DNA into single strands, allowing each strand to be copied. Helicase begins at a point in or at the end of a DNA molecule and moves one complementary base pair at a time, breaking the hydrogen bonds so that the double-stranded DNA molecule becomes two separate strands. DNA polymerase is another key enzyme in DNA replication. DNA polymerase adds nucleotides one by one to the growing DNA chain. DNA polymerase has two very important roles in DNA replication. It catalyzes the formation of covalent bonds between adjoining nucleotides in the growing DNA strand. It also serves in a proofreading role to ensure each new DNA strand is a near-perfect copy of the original. D1.1.4, amplifying and separating DNA. Polymerase chain reaction and gel electrophoresis as tools for amplifying and separating DNA. Students should understand the use of primers, temperature change, and Taq polymerase in the polymerase chain reaction PCR, and the basis of separation of DNA fragments in gel electrophoresis. The exploration and manipulation of DNA have been enhanced by the development of many different techniques during recent decades. In this section, we will examine two of these techniques: polymerase chain reaction, PCR, and gel electrophoresis. Polymerase chain reaction. PCR is a laboratory technique using a machine called a thermocycler, which takes a small quantity of DNA and copies all the nucleotides to make millions of copies of the DNA. See figure 5. A polymerase chain reaction, PCR, is a method of generating enough DNA for analysis. PCR is specific in that it only amplifies a targeted section of DNA. Various components are needed to run a PCR, including the following: primers, single-stranded short polymers of 15 to 20 nucleotides that are complementary to the nucleotides at one end of the target DNA to be copied. These primers provide a starting point for DNA synthesis because DNA polymerases can only attach new DNA nucleotides to an existing strand of nucleotides. Taq polymerase. Taq is a polymerase from a bacterium that lives in hot springs. This enzyme can withstand high temperatures. Free nucleotides to synthesize the new strands of DNA. Once all these components have been mixed in a tube, the tube is placed in the thermocycler, which controls the temperature of the reaction. There are 3 steps to the PCR process. Step 1. Denaturation. The mixture is heated to a temperature between 92 degrees Celsius and 98 degrees Celsius, which breaks the hydrogen bonds holding the two strands of DNA together. Step 2. Annealing. The mixture is then cooled to between 50 degrees Celsius and 65 degrees Celsius to allow the primers to bind with nucleotides on both strands at the ends of the target sequence. Step 3. Elongation. The Taq polymerase catalyzes the building of new DNA strands by extending the primers. A temperature of 70 to 80 degrees is needed for this step. This three-step cycle is repeated over and over until enough target DNA has been produced to accomplish the desired task. Gel electrophoresis. This laboratory technique is used to separate fragments of DNA in order to identify its origin. Enzymes are used to chop up the long filaments of DNA into varying sizes of fragments. If two different sources of DNA are to be compared, the same enzymes are used to produce fragments. The DNA fragments are then placed into small wells, or holes, in a gel, placed in an electrophoresis chamber. Figure 6 illustrates an electrophoresis chamber. An electrophoresis chamber with power supply and electrodes. Notice the buffer solution in the holding tanks at the ends of the chamber. An agarose gel with sample wells is placed on the chamber bed, the middle region of chamber between the end tanks. The gel, with samples of fragments in the wells, is exposed to an electric current, positive on one side and negative on the other. The biggest, heaviest, and least charged particles do not move easily through the gel, so they get stuck very close to the wells they were in at the beginning of the experiment. The smallest, least massive, and most charged particles pass through the gel to the other side with a little difficulty. Intermediate particles are distributed in between. At the end of the experiment, the fragments leave a banded pattern of DNA, like the example shown in Figure 7. Figure 7. This autoradiogram or audioradiograph shows banded lines that were formed from 9 different DNA samples during electrophoresis. The black traces are left by the radioactivity of the materials used in marking the DNA samples. Smaller chains of DNA travel farther on the gel than larger chains, allowing characteristic banding patterns for a particular sample of DNA. D1.1.5 Applications of amplifying and separating DNA. Applications of polymerase chain reaction and electrophoresis. Students should appreciate the broad range of applications, including DNA profiling for paternity and forensic investigations. Nature of science. Reliability is enhanced by increasing the number of measurements in an experiment or test. In DNA profiling, increasing the number of markers used reduces the probability of a false match. The process of PCR and DNA profiling have been used to sequence the bases of human DNA. This sequencing was the goal of the Human Genome Project. The process of matching an unknown sample of DNA with a known sample to see if they correspond is called DNA profiling. This is also sometimes referred to as DNA fingerprinting because there are some similarities with identifying fingerprints, although the techniques are very different. If, after separation by gel electrophoresis, the pattern of bands formed by 2 samples of DNA fragments is identical, it means that both must have come from the same individual. If the patterns are similar, it means that the 2 individuals are probably related. DNA profiling can be used in paternity suits when the identity of someone's biological father needs to be known for legal reasons. At a crime scene, forensic specialists can collect samples such as blood or semen which contain DNA. Often, such samples only contain very small amounts of DNA. In this case, PCR is carried out to amplify, means to create more, of the DNA available for profiling. Gel electrophoresis can be used to compare DNA collected from a crime scene with that of suspects. If they match, the suspect can be questioned further. If there is no match, the suspect is probably not the person the police are looking for. Criminal cases are sometimes reopened many years after a judgment was originally made to consider new DNA profiling results. In the USA, this has led to the liberation of many individuals who had been sent to jail for crimes they did not commit. Figure 8. These 7 tracks were produced by gel electrophoresis to allow investigators to analyze and match DNA samples. Using Figure 8, showing the DNA profiles from 6 suspects, can you identify which one matches the DNA profile of the blood stain found at the crime scene? I guess that would be the blood from the murderer, not blood from the victim. Wouldn't be much sense in matching blood from the victim to a suspect. DNA profiling is used in other circumstances too. For example, in studies of ecosystems, when scientists use DNA samples taken from birds, whales, and other organisms to clarify relationships. This has helped establish a better understanding of social relationships, migration patterns, and nesting habits, for example. In addition, the study of DNA in the biosphere has given new credibility to theories of evolution. DNA evidence can often reinforce evidence of common ancestry based on anatomical similarities between species. Nature of science. How do we decide when evidence is reliable or not? Often when DNA evidence is used in a courtroom trial, it has a certain credibility as scientific fact. Yet, we know from our own experience in laboratory work that there is a degree of error in any procedure. Whether it be in the laboratory or in a courtroom, it is difficult to imagine evidence that can be considered 100% certain. When a scientist comes up with new evidence, old theories can be challenged or even overturned. Reliability of data can be enhanced by increasing the number of measurements in an experiment or test. In DNA profiling, increasing the number of markers used reduces the probability of a false match. A marker refers to a particular sequence of DNA. D1.1.6 DNA polymerases. Directionality of DNA polymerases. Students should understand the difference between the 5' and 3' terminals of strands of nucleotides, and that DNA polymerases add the 5' of a DNA nucleotide to the 3' end of a strand of nucleotides. The bonds of DNA. Each strand of DNA is composed of a backbone of alternating phosphate and deoxyribose molecules. These two molecules are held together by a covalent bond called a phosphodiester bond, or linkage. A phosphodiester bond in DNA forms between a hydroxyl group on the 3' carbon of deoxyribose and the phosphate group attached to the 5' carbon of deoxyribose. Study Figure 9, showing the nucleotide. You can see that the carbons each are given a number, 1', 2', 3', so that you know which carbon it is in the molecule. Each nucleotide is attached to the previous one by a phosphodiester bond. This produces a chain of DNA nucleotides. So here in Figure 9, a DNA nucleotide is composed of a molecule of deoxyribose, pentose, with a phosphate group attached to the 5' carbon and a nitrogenous base attached to the 1' carbon. Notice the hydroxyl, the OH there, attached to the 3' carbon. Each time a nucleotide is added, it is attached to the 3' carbon end. Even when thousands of nucleotides are involved, there is still a free 5' carbon end with a phosphate attached and a 3' carbon end with a hydroxyl group attached. The reason the DNA chain is built in this 5' to 3' direction is that DNA polymerase can only add nucleotides to an existing chain in this direction. This creates the alternating sugar-phosphate backbone of each chain shown in Figure 10. Figure 10, 5 nucleotides bonded to form a very small section of a strand of DNA. Note the carbon numbers of the pentose and what is attached to the 1', 3', and 5' carbons. As nucleotides are linked together, a definite sequence of nitrogenous bases develops. This sequence carries the genetic code that is essential for the life of an organism. Antiparallel strands of DNA. The 2 sugar-phosphate backbones are attached to one another by their nitrogenous bases. The 2 backbones or chains run in opposite directions and are described as antiparallel. One strand has the 5' carbon on the top and the 3' carbon on the bottom. The other strand is the opposite way round. Study Figure 11. The antiparallel strands of DNA run in opposite directions. The hydrogen bonding between bases is discussed in Chapter A1.2. D1.1.7 Leading and lagging strands. Differences between replication on the leading strand and the lagging strand include the terms continuous, discontinuous, and Okazaki fragments. Students should know that replication has to be initiated with RNA primer only once on the leading strand, but repeatedly on the lagging strand. The first step in the DNA replication process is the separation or unzipping of the double helix by the enzyme helicase. Helicase accomplishes this task by breaking the hydrogen bonds connecting the 2 strands. The point at which helicase is working to unzip the DNA molecule is called a replication fork. DNA strands can only be assembled in the 5' to 3' direction because of the specific action of polymerase III. Because the 2 strands of DNA are antiparallel, there is a difference in the process of assembling the 2 new strands of DNA from the template. For the 3' to 5' template strand, the DNA strand is formed in a continuous pattern and is relatively fast. The strand produced in this continuous way is called the leading strand. The other new strand forms more slowly and is called the lagging strand. The formation of the lagging strand involves fragments and is discontinuous. This discontinuous replication requires another enzyme, DNA ligase. The replication process is as follows. See Figure 12. 1. The leading strand is assembled continuously towards the progressing replication fork in the 5' to 3' direction. 2. The lagging strand is assembled by the production of fragments moving away from the progressing replication fork in the 5' to 3' direction. 3. The fragments of the lagging strand are called Okazaki fragments after the Japanese scientist who discovered them. 4. Primer, primase, and DNA polymerase III are required to begin the formation of each Okazaki fragment of the lagging strand and to begin the formation of the continuously produced leading strand. 5. The primer and primase are only needed once for the leading strand because it is produced continuously. 6. Once the Okazaki fragments are assembled, an enzyme called DNA ligase attaches the sugar phosphate backbones of the lagging strand fragments to form a single DNA strand. Replication of a strand of DNA has to be initiated with RNA primer. RNA primer is only needed once on the leading strand because it is continuously formed. However, the lagging strand does not form continuously, and RNA primer has to be added repeatedly as the double helix of DNA is opened by the action of helicase. D1.1.8 Functions of specific enzymes and molecules. Functions of DNA primase, DNA polymerase I, DNA polymerase III, and DNA ligase in replication. limit to the prokaryotic system. Table 1 provides a summary of the enzymes and other molecules involved in prokaryotic cell DNA replication. It is important to note the functions of DNA polymerase 1, DNA polymerase 3, and DNA ligase, but they work in conjunction with the other enzymes to result in the successful replication of DNA. Helicase unwinds the double helix at replication forks. DNA primase synthesizes RNA primer. RNA primer marks the beginning of the replication process. DNA polymerase 1 removes RNA primer at end of replication and replaces it with DNA nucleotides. DNA polymerase 3 synthesizes the new strand of catalyzing the addition of nucleotides in a 5' to 3' direction. DNA ligase joins the ends of DNA segments and Okazaki fragments. Single-stranded binding protein keeps bases from reforming hydrogen bonds between them after helicase action. D1.1.9 removing mismatched nucleotides. DNA proofreading. Limit the action of DNA polymerase 3 in removing any nucleotide from the 3' terminal with a mismatched base, followed by replacement with a correctly matched nucleotide. DNA polymerase 3 has one other very important function in the replication process. It checks for accuracy in the daughter strand it is synthesizing. It can recognize mismatched nucleotides and remove it from the 3' end of the daughter strand. After the removal of a nucleotide mismatch, it reverses direction and resumes the synthesis of the new strand. The result of this very efficient proofreading is an error rate in bacteria of only 1 in 100 million base pairs. DNA replication in eukaryotic cells has many points of origin along each linear chromosome. At each origin, a bubble forms from which replication occurs in both directions. Eukaryotic chromosomes have ends composed of short sequences of DNA repeated over and over. These ends are called telomeres, or telomeres, and they require a special enzyme called telomerase for their replication. Guiding question revisited: how is new DNA produced? In this chapter, we have discussed how DNA replication is essential to produce new cells, both in unicellular and multicellular organisms. Semi-conservative replication results in every new copy of DNA possessing one strand from the parent DNA and one new strand. Complementary base pairings in the DNA replication process ensures a high degree of accuracy. Helicase is required to unwind and break hydrogen bonds so that the process of DNA replication can occur. DNA polymerase controls the addition of free nucleotides to the developing DNA strand. Because of the directionality of DNA replication, one strand of DNA forms faster than the other. How has knowledge of DNA replication enabled applications in biotechnology? In this chapter, we have discussed how polymerase chain reactions, PCR, are used to amplify DNA segments so that valid analyses can be carried out. A thermocycler, Taq polymerase, primers, and free nucleotides are necessary for the process of PCR. Gel electrophoresis uses an electrical field and agarose gel to separate segments of DNA. PCR and gel electrophoresis are often used together in attempts to solve crimes where small amounts of DNA have been recovered. Gel electrophoresis is also used in the study of ecosystems. Paternity questions can often be solved using gel electrophoresis.